And now it's my pleasure and honor to introduce Dr. Stephen Archer, who is the head of the Department of Medicine at Queen's University. Dr. Archer is the head of the Department of Medicine and holds a C. Frankly Helene Key Bracken Chair. He is a physician scientist and a graduate of Queen's University. He is practicing cardiologist who specializes in care of patients with various forms of pulmonary hypertension. His other interests include strategies for improving cardiovascular care and training the next generation of physician scientists. In his spare time, he enjoys spending time with his family, and he's avid in hockey player, a guitarist, and a coffee drinker. I also like coffee drinker. We share that one. Dr. Archer directs a CHR-funded research lab. He investigates the role of mitochondria, both as oxygen sensors and regulators of cell proliferation, and apoptosis, and more recently, as participants in the lung injury in COVID-19. He has over 300 publications, and his age index was 103, with approximately over 41,000 citations. Impressive. Congratulations, Dr. Archer. His research focuses on exploring cellular and molecular mechanisms of oxygen sensing and mitochondrial metabolism and dynamics. He also develops experimental therapeutics for pulmonary hypertension and cancer. He has mentored over 70 trainees, many of whom are leaders in science and medicine. Dr. Archer is the founder and scientific director of Queen's Cardiopulmonary Unit, which is over $8 million and 8,000 square foot research facility, which has an approach from bench to bedside philosophy. His translational cardiovascular research has been recognized with numerous awards, including being elected as a Fellow of the Royal Society of Canada and has been awarded the Distinguished Scientist Award from the American Heart Association and American College of Cardiology. He received the AFCMC President's Award for Exemplary National Leadership in Academic Medicine and was named Chicago American Heart Association Cour d'Or recipient in 2013 for leading the establishment of a pre-hospital STEMI care network in Chicago. Dr. Archer, it's a pleasure to have you here. The floor is yours. I believe you're muted, Doctor.
Okay. Maybe if I unmute myself after 2.5 years of COVID, can you hear me now? Is that okay?
Yes, we can.
Okay, well, thank you, Dr. Andrea. So that was an overly kind introduction, and I'm probably going to disappoint you 100% after all the nice things you said, but I'll try not to. So what I'm going to tell you today briefly is that mitochondria are a major target, as we heard in the nice introductory part of the session for the SARS-CoV-2 virus and other coronaviruses. And that explains that this attack on the mitochondria explains many features of the disease. This is just an interesting picture from my lab taken by Jeff Mewburn showing 3 pulmonary artery smooth muscle cells with red mitochondria and blue nuclei. So just a reminder, science can be fun and artistic. So this work would not have happened without a huge team. And I'm sorry, I don't have time to introduce these people who come from University of Saskatchewan, University of Toronto, and Queen's University. And they include virologists, basic scientists, graduate students, technicians. It's an amazing group of people, and they were all so incredibly proud to do this work. They came to work every day during the pandemic. In fact, our lab was only closed for 3 weeks out of this entire time. Otherwise, people have been at the bench doing this work. So my objectives today are basically to remind you that this is the 3rd coronavirus epidemic we've had in 20 years and better figure it out for the next one, SARS, MERS, now COVID-19. I'm going to show you a very brief case, remind you that the major toxicities of coronaviruses are cardiopulmonary, that current COVID therapies, although they work, are not curative in many cases. Paxlovid has certainly been a major addition to the armamentarium recently. And also, I think the thing that's shocking, like a science fiction novel, is that at least 20% of people would not take an effective vaccine, no matter how encouraged they were to take it. So it will be helpful to have other strategies to treat these conditions. So the new idea that I'm going to present today is that mitochondria and airway epithelial cells and in pulmonary artery smooth muscle cells are targeted by the SARS-CoV-2 virus and other viruses. And the consequences are that the mitochondrial damage contributes to the airway infiltrates by causing airway epithelial cell apoptosis in large amounts, and contributes to hypoxemia by suppressing the lungs' normal autoregulatory mechanism, which is called hypoxic pulmonary vasoconstriction. And also that maybe enhancing HPV could have some clinical utility. So I probably don't need to show you this, but it is still staggering that by April 23rd, there were 508 million infections, and that's probably a gross underestimate by multiple fold. 6.2 million deaths, also probably undercounting, and global disruption of trade, which we all experience. And this is a case, and it sort of seems almost cute and historical. I used to work at the University of Chicago. We knew so little about this when it first started, but a friend of mine, Will Sharp, was an emergency doc and shared this very typical case, which got me thinking about the features of COVID early on in the pandemic. 60-year-old woman with a history of hypertension comes in with shortness of breath, cough, diarrhea, runny nose, sore throat, and abdominal pain. She'd been exposed to a coworker at the Illinois Department of Public Health 2 weeks before, and that's when she started to get sick. Her vital signs are listed there, the remarkable features being her tachycardia, 105 beats a minute, and a respiratory rate of 30 breaths per minute. And her O2 stat was only 75% on room air. And remember, she just walked into the emergency room, and the ER doc said she doesn't look that bad, as her oxygen saturation and tachypnea would indicate. And that sort of paradox was called happy hypoxemia, the disconnect between profound hypoxemia and the way the patient actually felt. And this is her chest x-ray. And for those of you that are not respirologists, I'll just point out the heart's in the middle, obviously that boot-shaped structure. And then on both sides, you can see the lungs, and they're diffused white out of the lungs, or alveolar infiltrate, sort of an ARDS, adult respiratory distress pattern. And I won't go through her lab values, but her oxygen level on a blood gas was only 68 millimeters of mercury, normal being above 90. And so she was enrolled on what was then a promising research study of remdesivir. Spoiler alert, it didn't prove to be very effective. She ultimately succumbed to COVID-19. So I just want to remind you that COVID-19 is caused by a predominantly droplet-borne virus, SARS-CoV-2. It's enveloped in a single-stranded positive sense RNA virus. And there have been 3 outbreaks, as I mentioned. And we're going to learn something from the SARS, the virus that causes SARS, in a moment. And there's a lot of lung injury in this condition, diffuse alveolar damage, abbreviated DAD, and a disproportionate hypoxemia, more hypoxemia than you'd expect from the infiltrate. And that's due to ventilation perfusion mismatch. And I'm going to explain hypoxic pulmonary vasoconstriction to you in a moment. The mortality rate on average is 1.5%. It varies by strain and by country. 5% of people are hospitalized. So this is the enemy, the virus, and I think most of you know it's got about a 30 kilobyte genome, which encodes 16 non-structural proteins that are called NSPs, and they each have a number. And then there are 4 structural proteins, which you're even more familiar with because you've all heard of the spike protein, which is what our vaccines are targeted. And then there are 9 accessory factors. And the spoiler alert on this is these proteins target proteins in mitochondria and interfere with their function. I won't spend too much time on this, but at the very beginning, and when I saw that case of Dr. Sharp's, I began to speculate on what the mechanism was of this happy hypoxemia and disproportionate hypoxemia. And so one of the things that we reminded our colleagues of in the literature was how the lung circulation normally works. And I won't spend too much time on this, but you can see the blue pulmonary artery with perfusion of the nice capillaries, and oxygenation occurs partway through the capillary bed. And the perfusion pressure is low, about 15 millimeters of mercury. And then the oxygenated blood emerges in the pulmonary vein. But if you have a green mucus plug, or if you have pneumonia in your airway, like you can see in this cartoon, the blood is diverted, the artery constricts, it goes to that particular alveolar segment, and it diverts blood, as you can see from the larger white arrow on the bottom, over to a healthy alveolar segment. That's called hypoxic pulmonary vasoconstriction, and it avoids ventilation perfusion mismatch. It puts the perfusion where the ventilation is. So if you get rid of HPV, you have a problem. One model of understanding COVID was that it was like ARDS, but in ARDS, there's marked pulmonary hypertension. So now you can see the blue arteries coming into the alveolar segments are constricted and small, and the pulmonary artery pressure is high. I've made these numbers up. 35 millimeters of mercury. The numbers are made up based on my understanding of the literature. But the real problem in ARDS is alveolar permeability. So there's a breakdown in the integrity of the alveolar wall with inflammatory cells. Another model that was out there at the beginning of the pandemic was this was like high altitude pulmonary edema, like you might get climbing Mount Everest. And high altitude pulmonary edema is different than what COVID is. In fact, in high altitude pulmonary edema, you see the right-hand alveolar segment in the bottom cartoon with a tightly constricted inflow pulmonary artery, but also tightly constricted pulmonary veins coming out of the alveolar segment. So you get both pulmonary hypertension coming in and pulmonary venous congestion going out, and that causes frothy alveolar edema with hemorrhage. So different than COVID-19. And what we speculated COVID-19 was a suppression of HPV, such that when you had pneumonia in the airway, you didn't get the constriction of the arteries. And you can see in the right-hand bottom quadrant, the arrows are luxuriantly perfusing the pulmonary segment, even though there's pneumonia there. And you're getting some of the features of ARDS with some alveolar leak. So the hypothesis that we're studying is that coronavirus proteins target mitochondrial proteins in airway epithelial cells, causing opening of the mitochondrial transition pore, and that causes excessive apoptosis, which causes lung injury and hypoxemia. Meanwhile, in the pulmonary artery cells, another cell in the lung circulation, they damage the mitochondria there. And here we postulate there's interaction with the electron transport chain, which is the site of the oxygen sensor. And that's 30 years of research, which I don't have time to summarize, but we've narrowed the oxygen sensor down to a component of electron transport chain complex 1 called induced 2. And we think it interferes with that. So again, just to make sure this is a crystal clear idea, the coronavirus binds to its spike receptor, the ACE receptor, comes into the cell on the left-hand side of this cartoon. It makes a viral polymerase so it can replicate itself, and then it goes to the ergic, and then it makes new virus particles, and then out of the cell and on with the infection. However, what we're saying, and I'll show you the evidence for it, is that some of those proteins go right to the mitochondria and target key mitochondrial proteins, either triggering apoptosis or impairing HPV, hypoxic constriction. So I'll show you some evidence for that in a second. But again, the cartoon here, just to show the viral proteins opening the mitochondrial transition pore, that depolarizes the mitochondria. And when the mitochondria depolarizes, it causes a leak of apoptotic factors like cytochrome C, the green balls, and apoptosis inducing factor, which was shown as blue balls here. And then in the panel C, you can see hypoxic pulmonary vasoconstriction is normally a mechanism involving complex 1 producing radicals, which are signaling molecules, and the virus, we believe, interferes with complex 1 function. So the consequences of this dual assault on mitochondria is airway epithelial injury, impaired HPV, causing alveolar leak, and increased VQ mismatch. So I probably have excessively introduced that, but it'll make the data simpler. So I just remind you that mitochondria do lots of things, and I'm going to show you that these coronaviruses, not just SARS-CoV-2, but another human coronavirus called H-CoV-OC43 and a mouse coronavirus called MHV1, interfere with all these functions. So the first function of mitochondria, as was said by prior speakers, to be the powerhouse of the cell, and ATP production is basically substantially inhibited by coronaviruses. But also mitochondria have this interesting non-canonical set of functions in which they join together. And as you read this, starting at the top, mitochondria fuse, and these fused mitochondria are more energetically efficient. And then the endoplasmic reticulum marks a spot in the mitochondria for fission or division. And then a protein called DRIP1, dynamine-related protein 1 that we study in our lab, makes a large ring, a macromolecular structure by binding its binding partners at that site and divides the mitochondria. That can serve multiple functions. Pathologically, it can release ROS, as was mentioned. It can also trim off bad parts of mitochondria for mitophagy, a quality control mechanism. And if it also, in dividing cells, is important to coordinate mitochondrial division with mitosis. That's called mitotic fission. So mitochondria do a lot of things, and they are actually known to be targets of viruses. So it's known that viruses will target mitochondria. And this is the mechanism of HPV, and this is the final introductory slide before the data slides. So if you look at the top right-hand corner, you can see the blue part of the lung. Pretend that's the part of the lung infected with COVID-19. And you can see that the arteries to that lung are supposed to be constricted, little tiny blue arteries, so the blood is diverted to the better ventilated lobes. And the way that's happening is shown in the cartoon on the left below it, which is that complex 1 is making superoxide radicals. The superoxide radicals get turned by SOD2 into hydrogen peroxide, which diffuses to the membrane, controls potassium channels, which regulate membrane potential and thus control calcium influx by the L-type calcium channel. And in disease states like COVID-19, there's damage to that complex 1 mediated oxygen sensing mechanism. So I'm going to show you data from 3 viruses, SARS-CoV-2, which requires a level 3 facility, collaboration with University of Saskatchewan and Dr. Banerjee and the VITO facility. HCoV OC43, which is a human pathogen, also coronavirus, collaboration with Jay Colpitts at Queen's, and MHD1, a collaboration with Gary Lavie at University of Toronto. So here's the data showing airway epithelial apoptosis. So the first thing, it starts with SARS, which is the disease that preceded SARS COVID-19. And this is a paper by Lay, and it's our remining of his data. And it basically shows what are the proteins, when you do a non-directed search, that are most regulated by this coronavirus infection. And as you read the right-hand bar graph, you can see it's mitochondrial matrix, ATP processes, cellular respiration, aerobic respiration. So even SARS, which we didn't think much about in terms of mitochondria, to my knowledge, and he wasn't focused in his paper on mitochondria, turned up massive regulation of mitochondria as one of the most disruptive pathways caused by SARS-CoV, the virus that causes SARS. So there's precedent. Then there's this exciting paper by Gordon that you're probably all familiar with. They took and cloned and expressed 26 of the 29 SARS-CoV proteins in human cells to see what would they interact with. They also looked for druggable targets, which I won't talk much about. But when we looked at their paper closely, there were several mitochondria interactome targets, which is quite exciting. And this is how they did their work. They took their SARS-CoV-2 proteins, which is the red diamond in panel A, and they looked at all the human proteins that interacted with. And if you look at the heat map in panel B, as selected in the red box, the mitochondrial matrix was highly regulated by many SARS-CoV-2 proteins. You can see the proteins by name on the bottom axis. But I won't, however you look at the details, the main point being, along with cell cycle regulation, et cetera, mitochondria were highly targeted by SARS-CoV-2 proteins. And the importance of this that I want you to reflect on is the fact that there's no virus here, just one protein. And although a virus never just gives you one protein during an infection, this shows you that it's protein-protein interactions rather than some inflammatory state that is capable of targeting the mitochondria. Interestingly, they found that these targets, the interactome was richest in the lung, which fits with the fact the lung is the major target, but it's also present in other cells, including the heart. And there's much more in this paper. This is just the interactome, and I've taken some of their data and replotted it. Panel B, you can see the M protein, which is a SARS-CoV-2 protein, interacts with many proteins involved in mitochondrial metabolism, which are the blue circles. Likewise, NSP8 interacts with ribosome. The ORF9C interacts with electron transport, as does NSP7. And you could go on with this, showing the protein-protein interactions. So does it actually do something? Well, we did whole cell transcriptomics on airway epithelial cells at 0 to 12 hours, work by Dr. Banerjee. And when you do an unguided, unbiased view of what pathways are most disrupted, as we saw from the previous speaker, you can see the mitochondrial matrix pops out as being the second most disrupted pathway or regulated pathway within 2 hours of a SARS-CoV-2 infection of an airway cell. And if you then, I know you can't read all this, but when you say, well, what is affected within the mitochondrial matrix family of proteins, and in this case, transcripts, you can see there's over 200 genes that are regulated within 2 hours, and the ones that are up are in green, and the ones that are down are in red. And I realize you can't read each of these, but I've blown some of this up to look at the fact that most of these, or many of them, are related to apoptosis. And in panel A, again, a little hard to read because of the font size, you can see the most regulated thing at 2 hours after SARS-CoV-2 infection in human airway epithelial cells is regulation of apoptotic signaling, with 200 genes regulated. And if you really have a myopic view of the world, you can actually read all these cool regulators, most of which you and I will never have heard of before this presentation. I'm not going to read their names. But we dug out some incredibly interesting apoptosis regulators that are upregulated in this early stage of the SARS-CoV-2 infection. While we were doing this, of course, it's a competitive field. We're still waiting for our paper to get published. Ramachandran published a paper that basically shows the same thing. This is an unguided whole transcriptome look at what are the pathways that are involved in PBMCs. His paper has a more cardiac focus. Ours is more lung focused. Ramachandran is showing that in peripheral blood mononuclear cells from patients with SARS-CoV-2, very small numbers, that the most regulated pathways, as you can see from the red bar graph at the bottom with fourfold regulation, are mitochondrial ETC complex 1 assembly and mitochondrial ETC NADH ubiquinone function. So fits, our work fits with his work fits with ours, and the mitochondria is a target. Here's some work using H-CoV OC43 that we did in our lab. We first, in panel 1, confirmed the infection of H-CoV OC43 with a nucleocapsid reporter. And I won't take you through each Western, but if you read, the first column is always the control, and then the infected column is next. And you can see upregulation of DRIP1, the mediator of mitochondrial fission, downregulation of CDK4, the mediator of cell cycle progression, and then a whole bunch of apoptosis mediators are upregulated like SMAC Diablo, caspase 1, BID, and AIF. And if you look at the bottom right-hand panel, it's quite fascinating. Look at the bottom right-most of those pictures. You can see in green, we show you the virus. That's the H-CoV OC43 virus. In yellow, we're showing you apoptosis inducing factor on the surface of the mitochondria, which is cut like a sausage in cross-section in red, and the nucleus is in blue. So it shows the virus intimately associating with apoptosis proteins on super resolution microscopy on the surface of the mitochondria. And then MHV1, which is the mouse virus. And you can see here, these are murine airway cells and with mitochondria in red. And this is TMRM, tetramethylrhodamine. The more red, the more hyperpolarized than normal the mitochondria. And look at what MHV1 infection does within 24 to 48 hours. It really markedly, as the theory predicts, depolarizes the mitochondria and they become less red. And as we saw in the human, the H-CoV OC43 infected cells, if you look at apoptosis inducing factor in the middle panel in green, you can see that there's a marked upregulation in the lower row, which is the infected cells that have much more AIF and some that's moved to the nucleus, which is an important step in apoptosis. So this is evidence that another coronavirus also turns on apoptosis by depolarizing mitochondria. Then we did experiments in vivo. This is a model I give credit to Gary Levy for inventing a number of years ago. We gave nasal instillation of the virus into mice, A6 mice, and you can see on the CT scan in the uninfected mouse, it looks very much like a human CT scan. The histology shows nice normal alveoli, and the tunnel stain, tunnel being a measure of apoptosis in green, is negative. There's no green nuclei. Then when we cause MHV1 pneumonia, you can see the lobar consolidation of the right lung on CT scan, the inflammation in the airways on the right histology, and then lots of tunnel positivity. So we're getting induction of apoptosis not only in cells, but also in vivo. And this is true also in another model work by Chan's lab using the SARS-CoV-2 virus itself in the Syrian golden hamster. And you can see the green nuclei in the tunnel assay on the bottom right-hand side there shows that apoptosis is in fact a part of this disease in animal models. And is it enough to we just have certain proteins? So we picked the proteins that we knew from Gordon's work targeted mitochondria, and we gave just one protein at a time to airway epithelial cells to see if we could turn on apoptosis without the virus. And as you can see here, AIF, I'll just focus on that. The NSP7, NSP9, and M protein all upregulate the expression of AIF relative to the GFP control. And they also regulate DRIP1, the mediator of fission. So this can happen even without infection. I won't focus on, I think for time's sake, some of these proteins will also block the cell cycle, but I'm going to skip that. This is Ramachandran's paper that I mentioned, and what Ramachandran did that we have not yet finished doing but are working on is he actually did immunoprecipitation and showed that these viral proteins, look at the black circles in the first part of the cartoon, actually physically interact with portions of the mitochondrial transition pore, and there is protein-protein interaction that is resulting in that pore opening, which is the first step in depolarization and apoptosis. We have preliminary findings similar to this. These are in cardiac cells. And again, while we were waiting for our paper to be peer reviewed, another paper came out also showing that this B cell related ovarian killer protein BACC is regulated by SARS-CoV-2 and it results in apoptosis. So there's increasing evidence from other labs as well that apoptosis is indeed induced. What about mitochondrial fission? Well, this is some cool work from our lab done in collaboration with Vito and Saskatoon. You can see the mitochondrial network in an airway epithelial cell in red. It's nice and fused as it should be. And then when we infect with SARS-CoV-2 in the bottom panel for 72 hours, it's fissioned, fragmented mitochondria. We do machine learning on that, which allows the computer to actually count the size and shape of mitochondria and make a bar graph. And you can see in the columns on the right that the punctate mitochondria in red, there were very few of them at the beginning. And then with the infection, there's marked upregulation of punctate mitochondria and marked loss of the filamentous mauve mitochondria. So yes, these viruses cause fission, and they do it in endothelial cells. These are human pulmonary artery endothelial cells. Same thing exactly happens. Control on the left, the fissioned mitochondria of an infected PA endothelial cell. And whether this relates to the thrombotic predisposition, I'm not sure. Here's some electron microscopy with H-cov OC43 showing the mitochondria becomes swollen. And you can see the quantification in the bar graph, and you can actually see the viruses accumulating within the cell, the little typical coronavirus-shaped particles within vesicles within these airway epithelial cells. Here's some super resolution microscopy looking at the mock infected versus OC43 infected cells. And again, you can see fragmentation. The green is apoptosis inducing factor. And you can see how the mitochondria become fragmented and swollen as they lead towards apoptosis. And it's enough to produce fission to simply give the right protein. Not all viral proteins cause fission, but in this slide, the controls in the top left, and you can see that NSP7 and NSP9 transduction, we used a lentiviral vector for them, causes fission compared to the nice fused mitochondria and epithelial cells on the top left-hand panel. So many of these functions of the infected particle itself are replicated by the viral protein in isolation. And this is Ramachandran finding the same thing in the top panel E. You can see the fragmented green mitochondria in his paper. So what about bioenergetics? We saw some cardiac work at the beginning of the session, and basically we find there's a marked depression of, this is seahorse data looking at the control energetics in airway epithelial cells versus those infected in red with OC43. And you can see the baseline oxygen consumption rate in the top panel is reduced, and there is no respiratory reserve at all. You see in the blue line, when you give FCCP, you get a big increase in respiration, which is normal, and that's completely obliterated by infection. And complex 1 activity in the bar graph on the right is down. So as predicted, there's marked bioenergetic impairment. And for time's sake, I'll just say, and it's exactly the same with MHV1. You can see the difference between control in white and the MHV1 infected in gray here with marked suppression of oxygen consumption, especially respiratory reserve. And there's also an increase in glycolysis as measured by ECAR. So it causes apoptosis, causes fission, impairs bioenergetics. And again, for time's sake, I won't spend much time on this, but NSP7 protein does the same. And you can see a suppression in the white squares of oxygen consumption rate at baseline in the top left panel, and also of the respiratory reserve in the third section of the OCR graph at top. And this is showing that Ramachandran found the same thing, although he found it with the M protein. So this is a seahorse trace that Ramachandran published in panel A. And look how depressed the oxygen consumption rate is in the green with the M protein compared to the other proteins or the vector control, which is black. And respiratory reserve capacity is also obliterated. Whether this causes long COVID or not, but I think it's interesting. And then the last couple of minutes is just showing you the impairment of HPV, the VQ matching system. So these are human pulmonary artery smooth muscle cells. And basically the first step in hypoxic constriction, as I told you, is the membrane depolarizes and calcium rises. So we measure intracellular calcium as a surrogate for HPV. And you can see in the rise in calcium in the representative trace in the middle, where you go from normoxia to hypoxia, cytosolic calcium rises, and this is followed by constriction. And you can see in the GFP control in the bar graphs, there's a nice rise in calcium, and in And there's also a nice rise in purple when you give KCL, which is independent of oxygen sensing. But if you infect with NSP7 or NSP9, you can see there is no M protein or NSP9, there is no rise in calcium in the pink bar, although the KCL still causes a rise in calcium. So both M and NSP9 are suppressing HPV in this cellular model. But if you don't like cellular models, this is pretty much the last data point. This is infection of mice that I showed you the CAT scans before. And you can see the baseline saturation in the male mice and the female mice is both down in the 70s, just like a patient. And then we did right heart catheterizations and we measured the rise in PA pressure in response to acute hypoxic ventilation. So in the panel 2, the control PA pressure is low, just like in a human, under 20. And then you make the mouse hypoxic, and it gets a nice rise in pressure. That's hypoxic pulmonary vasoconstriction. Then we give a calcium channel agonist called BAK8644, which enhances HPV, and we get huge increases in hypoxic pulmonary vasoconstriction, and those RV systolic pressures rise in the right-hand part of that upper trace. But look at the MHV1 mouse below it. There's no HPV at all. You go from normoxy to hypoxia. There's no rise in RV pressure. However, when we give BAK8644, we can restore HPV. And the thing that's important is when we look at panel 3, the bays and the pink bars, and we can see that there's a rise in O2 saturation systemically that occurs when we enhance HPV. So this could be a therapeutic strategy that you might use in MHV1 pneumonia. So we certainly are demonstrating loss of HPV. And very gratifying for the hypothesis that we published in 2020 was this mouse data that I just showed you, and also a very nice paper by Caravita, and they were also kind enough to actually cite our work, which is nice. Basically looked at patients with COVID-19, and they showed they had impaired HPV, that there was increased shunting related and low PVR, pulmonary vascular resistant, that was different than ARDS, and very much a case of impaired hypoxic pulmonary vasoconstriction. And for time's sake, I'll just show you the one slide. The COVID-19 patients are on the left column, and the right column is the control group. And QSQT is a measure clinicians use of shunting in the lung. So VQ mismatch or impaired HPV, higher numbers are bad. And you can see there was threefold higher QSQT in the COVID patients than in a group of dyspneic patients. And as predicted, the mean PA pressure was slightly elevated at 27 in the COVID patients relative to the control patients at 17, but the pulmonary vascular resistance, which is a measure of whether those arteries are constricted or not, was 0. None of them had increased PVR. So that would fit with the idea that this is an impairment of HPV, not like ARDS, not like high altitude pulmonary edema. So I hope I've convinced you in this talk that the virus comes into the cell, and in addition to replicating itself, it makes proteins that target the mitochondria by protein-protein interactions. Some of them open the transition pore and cause apoptosis. Some of them interfere with complex 1 and block HPV. And I think that while vaccination is very important, our fellow humans have shown an incredible ability to not be science-based and to avoid vaccines. So we're going to need other strategies. This is the third pandemic, so we need to better understand this. And I think mitochondria in blue there offer new therapeutic targets, and that's what we're working on. So with that, I'll end, and thank you for indulging me on this slightly overly long talk. I appreciate your attention.
Thank you very much. This was fascinating. I was not disappointed at all. By the contrary, it was fascinating. We are on the top of the hour, but I want to allow a time for 1 or 2 questions. I'm going to start with a question about mitochondrial inflammation. As part of SARS-CoV-2 is also being noted that inflammation, and you talk a little bit about that in your presentation as well. Do you see mitochondria be possible one of the causations of the inflammation seen on the SARS-CoV-2?
Yes, that's an excellent question. And we have a separate program in our lab studying the inflammasome. And one thing I know about that research that we've been finding as well is when you take macrophages and activate them, whether it's by a virus or other mechanisms, they basically will directly target mitochondria and cardiac myocytes. And I suspect that fission and fragmentation they cause is a source of the damps that turn on the NLRP3 inflammasome. So I think it is a vicious circle. Inflammation damages mitochondria, damaged mitochondria make damps, damps cause inflammation. So I think and it's interesting in the patients that we look after in my clinical life, the patients that do the worst are those that have excessive inflammation. So I think Yeah, and that's why dexamethasone is a helpful drug, one of the first helpful drugs in the pandemic. And so I think, although the virus can directly target the mitochondria, I think probably in real life, you get inflammation also targeting the mitochondria, and then the mitochondria driving the inflammation.
So potentially having a healthy mitochondria will help you to boost the COVID.
Yes.
That's fantastic. Again, thank you very much for being here. If we have additional questions, I will send directly to you. And I appreciate your time for joining us today, Dr. Archer.
Well, thank you very much. It's great to meet you guys all virtually, and keep up your coffee drinking, Dr. Anzer, as it's keep you alive and healthy.
I will. Take care. Bye now.
Bye-bye.